Click for IB Biology. Here we're looking at nucleosome structure, the interaction between DNA and the so-called histone proteins. There's a blank PDF of this presentation way down here in the description that's linked in, along with some questions that you can take. The answers are provided so that you can check your understandings. The end screen shows channel links and playlist links. When we see images of chromosomes in textbooks, they often take on this form here. Let's be aware that this is a combination of DNA and proteins, what is usually called chromatin. Now, the actual structure which is presented here will be dealt with when we do DNA replication, so we'll leave that till then. But when we explore the actual chromatin structure, we find that DNA is wound up into tighter and tighter coils. The ones that we're going to discuss here relate to the 10 nanometer fiber known as the nucleosome. So perhaps one could think of this as the simplest way in which DNA coils upon itself. There is an octamer of 8 histone proteins around which DNA winds itself. There's usually 1.75 turns, 147 bases included in that, and finally secured by a 9th histone protein known as the H1 linker. The DNA actually interacts with the histone proteins. Overall, we can see that DNA, because of the phosphate groups, is generally negatively charged. The histone proteins, like all proteins, will have an N group at one end, the N terminal, and a C at the other one, and this N terminal becomes positively charged. And so the positively charged ends of the histone proteins become attracted to the DNA molecule, and this affects the expression and regulation of genes, and actually gives us a link into the process which is known as epigenetics, to which we will also return in a later video. You can observe these N-terminal interactions with the histones by visiting the protein data bank. A link is provided in the description below, and when there, if you enter the phrase nucleosome core particle and then select probably the first box that comes up, you can then move forward with the link to selecting one or two of the links, one of the links which looks like this one here, and you will be able to interact with the molecules. The molecules can be rotated and changed around in various dimensions, exploring it in a 3D space by clicking on the image there. I've created a separate video which guides you through this process, and that will also be linked into the description below, so you can interact with the histones and the DNA in these molecular models. As is mentioned, many of these topics raised will come up in other issues such as mitosis and gene regulation, gene expression. Links here are to the final video in this nucleic acid structure sequence, to the channel, and of course to the playlist.